Here we are again with the latest news from the website, prostatewarriors.com.
Always great to be here.
Welcome, everyone, to another deep dive. We've got a very specific and I'd say incredibly important mission today.
We really do.
We are sifting through a massive stack of recent updates straight from the front lines of advanced prostate cancer research. So for you listening, we're going to tackle the clinical trials, first starting with the heavy hitters and moving into some really cutting-edge cellular therapies. And then we're going to wrap up with a truly remarkable case study.
Right, because the goal here isn't just to throw raw survival data at you.
Exactly. It's to help you understand the rapidly evolving landscape of precision medicine and targeted therapies. There is a tremendous amount of innovation happening right now. Okay, let's unpack this. We have a lot of dense medical terminology ahead, but the sheer volume of progress is genuinely hopeful.
It's a profound shift. And while this information is undeniably complex, understanding the why behind these treatments is empowering. Whether you're a patient navigating your own care or an advocate, knowing how these therapies work equips you to have better, more meaningful conversations with your oncology teams.
That empowerment is key. So let's look at the data actually driving that change. We'll start with the heavy hitters, the phase 3 clinical trials.
The large-scale studies that dictate the new standard of care.
Exactly. First up is the Capitello 281 trial. This is a phase 3 trial looking at a drug called Capivasertib combined with Abiraterone.
Right.
And this specific combo is aimed at patients with PTEN-deficient metastatic hormone-sensitive prostate cancer, or MHSPC.
PTEN deficiency is a subtype that affects a substantial portion of advanced cases. In simple terms, the tumors have lost a key tumor suppressor gene. They've essentially lost a vital brake pedal.
Which makes the cancer much more aggressive.
Exactly. But the numbers coming out of this trial are compelling precisely because they're targeting that missing brake pedal. They randomized 10,012 patients with strictly defined PTEN deficiency.
And half got the investigational combo, right? Capivasertib, Abiraterone, and standard androgen deprivation therapy.
Right. And the control group received a placebo with the standard Abiraterone backbone. The primary endpoint they were measuring was radiographic progression-free survival rPFS. The treatment group saw a median rPFS of 33.2 months, whereas the placebo group was at 25.7 months.
Wow. So you're looking at a clinically meaningful 7.5-month delay in disease progression.
Yes. And secondary outcomes heavily supported this new regimen, too.
But the real takeaway here isn't just the extra 7 months on a calendar. It's a crucial detail for patients. We are finally seeing aggressive combination therapies that don't force a patient to trade their daily quality of life for a longer lifespan.
That is huge.
It is. This trial placed a massive emphasis on patient-reported outcomes using the AFAS-ETP questionnaire, assessing functional aspects of daily life, pain levels, emotional well-being, the ability to work. And despite higher adverse events in the Capivasertib group, their quality of life held completely steady.
Roughly 80% of patients were able to preserve their daily functional abilities. Extending overall survival is the goal, but preserving the quality of that extended life is what makes a treatment viable.
And for those following the underlying science, this builds directly on preclinical synergies we've seen in the lab. It specifically attacks the pathway through CDK9 and actin these PTEN loss models.
It's highly targeted.
So if that trial is about targeting a specific genetic deficiency, how do we approach targeting the physical location of the disease?
That brings us to another massive phase 3 study, the PEACE 3 trial.
Right. This one shifts the focus to patients with bone-dominant disease, testing a combination of radium-223 and enzalutamide.
What's fascinating here is the precise mechanism of action at play. Radium-223 operates through a concept called calcium mimicry.
Because the bone metastases are rapidly pulling in calcium from the bloodstream to build scaffolding.
Exactly. So radium-223 mimics calcium on a molecular level. The tumor pulls it right in, thinking it's building material. But radium-223 is an alpha emitter.
I want to pause on that for a second because the type of radiation matters immensely here.
It matters entirely. Unlike beta radiation, which penetrates deeply, alpha radiation is like a microscopic bowling ball. It's heavy, it carries a massive amount of destructive energy, but it has a very short range, literally just a few cell diameters.
So it delivers highly targeted radiation directly to the bone metastases, sparing the healthy bone marrow.
Yes. And at the same time, the enzalutamide is actively blocking the androgen signaling that feeds the systemic progression of the cancer. You're dropping localized heavy artillery on the bone tumors while cutting off the fuel supply to the rest of the disease.
And the final data backs up that dual approach beautifully. Adding 6 infusions of radium-223 to daily enzalutamide extended the median overall survival to 38.2 months.
Up from 32.6 months with just enzalutamide alone.
Right. And it improved our PFS to 19.2 months. Experts do recommend pairing this with bone protective agents to manage the side effects, but the survival gains are undeniable.
It represents a very strategic evolution in how we sequence care for bone-dominant disease.
Let's pivot from those broad, standard-defining Phase 3 trials to the incredibly cutting-edge world of cellular and targeted therapies.
The earlier phase trials.
Exactly. These are the Phase 1 and Phase 1/2 trials, smaller patient cohorts, but the absolute bleeding edge of oncology. First up is the PARTHENON trial. This is a Phase 1/2 study looking at an oral drug called AZD4956.
Designed to block a specific protein called SMARCA2.
Right.
This trial is a perfect real-world example of synthetic lethality. We know that roughly 20 to 30% of metastatic castration-resistant prostate cancer cases have homologous recombination deficiency, or HRD.
Often from mutations in genes like BRCA1, BRCA2, or ATM.
Right. Simply put, these tumors already have a broken DNA repair system.
They lack their primary mechanism for fixing cellular damage.
Exactly, and we can exploit that weakness. To survive, these cancer cells rely heavily on a backup system, the SMARCA2 protein. AZD4956 completely shuts down SMARCA2.
So the cell can't repair its natural replication errors, and it just collapses.
It's a highly targeted double hit, and in this trial, they're also testing it alongside saruparib, a PRP inhibitor, to squeeze that weakness even further.
Now here's where it gets really interesting. We also have the SYS6043 Phase 1 trial, and I love the analogy researchers use for this one, the Trojan horse approach.
Yes, the antibody-drug conjugate, or ADC.
It targets a protein called B7H3.
B7H3 is fascinating because advanced tumors use it almost like a cloaking device to evade the immune system. It appears on the surface of 20 to 80% of advanced tumors, especially aggressive bone metastases.
And healthy cells almost never express it, which makes it a great target. So the antibody finds the B7H3 protein, binds to it, and the cancer cell actually swallows the antibody, pulling it inside.
That is when the trap springs. Once internalized, it releases a topoisomerase I inhibitor. That inhibitor jams the biological machinery that unzips the DNA, and the DNA literally snaps into microscopic pieces from the inside out.
It just triggers immediate cell death.
Incredibly precise.
Precision really is the defining theme here, which leads us directly into the AM006 trial. This is another Phase 1/2 trial focusing on HER2 positive metastatic prostate cancer.
We constantly hear about HER2 and breast cancer, but 20 to 30% of metastatic prostate cancers also overexpress this exact same receptor.
So this trial uses HER2 FPBMC therapy.
A marvelous cellular engineering. They harvest the patient's own T cells and arm them with a bispecific antibody. You can visualize it as a microscopic piece of double-sided tape.
One end grips the T cell to hyperenergize it.
Right, preventing immune exhaustion. The other end binds to the HER2 receptors on the tumor, causing rapid direct contact killing.
And it doesn't stop there.
No, these armed T cells also release a massive cascade of cytokines, sounding an alarm that recruits other immune cells to the tumor site. It creates a lasting anti-tumor memory.
The biological engineering is just staggering. And keeping with that theme of reprogramming the immune system, we have to talk about the PPSMA 101 trial.
A very important Phase 1 trial.
This is a CAR T-cell therapy trial for heavily pretreated patients. We're talking about men who had been through up to 10 prior standard treatments. Their options were virtually zero.
They utilized an autologous CAR-T product genetically engineered to target PSMA.
And the efficacy data was genuinely impressive. 21% of these patients saw their PSA levels decline by 50% or more.
With one patient dropping over 99%.
A 99% drop on an 11th line of treatment. But hitting the immune system with that much engineered force comes with a serious cost.
It does. We have to balance the efficacy with the reality of the side effects. 61% of the patients experience cytokine release syndrome, or CRS.
Severe systemic inflammation.
Yes, requiring extremely careful inpatient management. But they incorporated a brilliant fail-safe and ICASP9 safety off switch.
So if the toxicity gets too high, doctors can administer a drug that commands the CAR T cells to immediately die off.
Exactly. It effectively manages the toxicity.
So what does this all mean for actually finding the cancer? Because precision medicine inherently requires precision diagnostics.
You cannot target what you cannot accurately see.
Right. That brings us to our Phase 2 imaging trial, the Co-PSMA trial.
This study looked at a next-generation PE imaging agent called 64Q SAR-bisPSMA, testing it against the current standard 68GA PSMA-11.
And the new copper-64 agent was vastly superior. It more than doubled the mean lesions detected per patient, finding 1.26 lesions versus just 0.48 with the standard agent.
I want to address a psychological point for the listener here. Finding more lesions sounds scary. It triggers anxiety. But in the context of biochemical recurrence, finding more is actually a massive advantage because it means the tracer is highly precise. Exactly. It doesn't mean the cancer is more aggressive. It just means the medical team finally has a completely accurate map, allowing for earlier, more accurate salvage therapy.
And there's a practical benefit too. The copper-64 agent has a 12.7-hour half-life,
Which is a huge logistical improvement. It allows for next-day imaging. Giving the tracer 24 hours to clear out of the healthy tissues yields significantly better contrast on the PT scans.
It's like letting the mud settle in a pond before looking for a dropped coin.
A perfect analogy.
To wrap up our deep dive today, we're moving away from the large trials to focus on a single case study.
A rare outlier, but a very important one.
This was a heavily pretreated man with metastatic castration-resistant prostate cancer who achieved a complete durable response lasting over 4 years using the immunotherapy drug Pembrolizumab.
As a medical professional, I have to provide the impartial context here. This is an incredibly rare outlier. Prostate cancer is typically an immunologically cold tumor.
It doesn't usually respond to standard checkpoint inhibitors.
Right. But his tumor had a highly specific molecular profile. It was MSIH, it was mismatch repair deficient, had a high tumor mutational burden, and harbored a BRCA2 alteration.
All those factors at once.
Yes. If we connect this to the bigger picture, the probability of all those features lining up is well under 1% of advanced prostate cancers. But because of that rapid mutation, the tumor was highly visible to the immune system once the drug stripped away its cloaking mechanisms.
And that perfectly highlights why broad molecular profiling is so essential for patients, because finding these rare anomalies can unlock life-saving immunotherapy.
It proves the fundamental thesis of precision oncology. We cannot just treat the statistical average. We have to actively look for the exceptions.
Which brings everything full circle for you, the listener.
Yeah.
Why all this dense medical data matters. Whether it's a phase 3 trial extending survival while preserving your daily life, or a rare case study proving the immense value of genetic testing, the overarching theme is that prostate cancer treatment is becoming highly personalized.
Highly personalized and incredibly precise.
Knowledge of these trials equips you to sit down with your oncology team and have proactive, highly informed conversations about the best path forward for your specific biology.
And I'll leave you with a broader concept to ponder. Building on all these targeted therapies, molecular profiles we've discussed today, what if the future of oncology isn't about treating prostate cancer at all?
What do you mean?
What if, in the near future, trials are grouped entirely by the unique genetic barcode of the tumor, regardless of where it started in the body? A prostate cancer patient sitting next to a lung cancer patient receiving the exact same targeted drug because their tumors share the same broken machinery.
That complete redefinition of the oncology community is the true horizon of precision medicine. We have covered a tremendous amount of ground today, and we hope this deep dive has given you some valuable clarity. Stay strong and fight on.